Okay, so here we have figure 3 of the Stoeger et al. Paper. In figures 1 and 2, there was basically some data suggesting in mice and in other vertebrate non-humans, this association between transcript length and a reduction in expression as those different organisms got older and older. And what Stoeger et al. Wanted to do next was basically see if they could find a similar pattern in humans. So what they did was they took RNA sequencing data from GTEx, which is this pretty massive initiative to take postmortem tissue from different humans, human samples, basically tissue separate them and sequence them with RNA sequencing to get an idea of the RNA expression in different tissues. So they did a very similar experiment to their mouse. To their initial mouse experiment, and they basically stratified the GTEx samples that they investigated into 3 groups: young, which were between 20 and 40 years old, middle age, which were between 40 and 60, I believe, and then older age, which were between 60 and 80 years. And they did the exact same gradient boost approach to characterize the same 2,300 or so features for their importance in really predicting how expression has changed as people age. And with GTEx, it's pretty exciting because you have so many more samples, which gives you a lot more power. So what did they find? Here we have panel A, which is the exact same sort of stacked box plots that we saw in figure 1. And what we're finding is that the features that are associated with the gene's length, like transcription length or coding sequence length, tend to again be the best predictors for how expression has changed with age. And in this case, gene length was the strongest, but for the rest of the figure, they wanted to stick to transcript length because that's what they had looked at in figure 1. So here we have in panel B, we are looking at just how strong that anti-correlation is with humans. So here we have middle-aged compared to the younger sample set and old age compared to the young sample set. And these box plots are showing the length correlation association between fold change and length compared to controls, which were a permutation experiment where they took all those samples and essentially just assigned them to some random pairing of the age groups. And what we see is consistently that almost all the tissues that they looked at have this anti-correlation. And what they found in their data set actually was that the most significant tissues were brain tissues, which we see in these brown box plots, which are sort of much more anti-correlated, which was a pretty exciting finding that they had. And I don't know if they didn't discuss looking at any sort of post-mortem implications of the tissue. I'd seen previously that there were some findings that brain has some pretty severe post-mortem changes that can really affect the transcriptome, and they didn't discuss that. Here is one example of the brain tissue they looked at, which was the frontal cortex, and where we can see with x-axis showing log 10 transcript length of a specific RNA sequence, or of a specific gene, sorry. And in the y-axis, we have the relative fold change between those different age groups to the youngest age group. And we can just see pretty clearly that there's this anti-correlation. As we have longer genes, we tend to see less expression with age. This was in the frontal cortex. And then the last thing they wanted to show here was basically comparing all of the tissues in these different matched human age groups. And what we're seeing is relative to the model organisms that there is this very similar pattern of most of the tissues having this long genes decreasing with age, and sort of there being a few that actually have an increase of expression with age. So it just goes to show that humans seem to fall in a very similar expression profile with the other non-vertebrate organisms.